All right, dear students, we will pick up where we left yesterday. Good morning. So we talked yesterday about why the role of the cell divisions and how monocellular organism divide regarding multicellular organism. They develop from a fertilized cells. Cell division also promote growth and repair. And during the cell division, the chromosome, which is the filamentous DNA, divided or replicate into two chromatid sisters that they still attached by the centromere. All cells in our body go through what we call cell cycle. 90% of the cell cycles is interphase. All the cells prior any division, either somatic cells or sexual cells, have to go through interphase, which is 90% of the cell cycle. You have three phases: G1 gap 1, stage S, so phase S, and gap 2. G1, anything inside of the cell is going, during all these phases, the cells grow, right? But during the G1, any structure that is inside of the cell, from mitochondria to chloroplast to ribosomes to Golgi, any structure will be replicated except your chromosome, your DNA. The DNA will be replicated during the S phase. During the G2, it's a phase that will control that everything is ready to enter the M phase. The M phase is the mitosis phase. It represents 10% of the cell cycle. And the mitosis, we have different phases also. We have a prophase. During the prophase, as you can see here in this picture, let me put my little hand. The nucleolus disappear. It's gone. And the chromatin is condensed. You remember what is chromatin? It's your chromosome plus protein, because during the division, a lot of factors, a lot of molecules, a lot of proteins are coming to help with this division. So we are no longer going to call it chromosome, but chromatin, right? They are condensed, and very, very nicely, the mitotic spindle began to form. And what is the mitotic spindle? I explained it to you very well yesterday. If you remember the centriole are 9 plus 3 microtubules. They are made of microtubules. They are like, they are going to move, and when they move, make like those spindles. It's like a web, a spindle. They are moving, and those spindles are only microtubules. So those mitotic spindles are beginning to form more and more. And those pair of centriole, they are going to start moving to the opposite pole. And by the way, during the G2 phase, the centriole start to be separated. And they keep being separated from each other. Because remember, centriole are being replicated during G1, because all the structures except the DNA are going to be replicated in the G1. Nothing is separated. chromosome are chromatid sister, everything's are together, except the centrioles start to be separated during the G2 phase. And during the prophase, they continue to be separated, forming this mitotic spindle. During the pro-metaphase, you are going to see it very, very well, those chromatid sisters. They appear very nicely. And one thing that you notice, the white things over here, this is the nuclear envelope. Nuclear envelope disappeared. We can still see some fragments, but completely is gone. And we saw that the centrioles are in the opposite side. That's it. They move where it's supposed to be. And we saw those microtubules, those webs between parentheses. Okay? We have, if you notice, and we went through this, we noticed two types of microtubules over here, of the spindles microtubules. We have one that they are over here, not attached. We have one that they are attached over here. You see, you remember what we have here. This is the chromatid sister attached by centriole. This is the centriole for each chromatid sister. And it seems like they are attached to the centromere, sorry. Oh, God. Centromere. It seems like they are attached to centromere. I'm not. This is a molecule over here that we call kinetochore. Kinetochore. So they are attached to those kinetochore. Look at those. Those microtubules are attached to kinetochore. Those microtubules attached to kinetochore. Those two, those are not. They're not attached to kinetochores. They are not. They're not attached to kinetochore. So therefore, we have two types that are visible. Non-kinetochore microtubules and kinetochore microtubule. And those are fragments of nucleus. Some fragments appear. This is the pro-metaphase. What's going to happen during the metaphase? That's an early metaphase over here. Pro-metaphase. It could be also a late prophase. Okay? During the metaphase, you are not going to miss it. You are not. Look at the confocal microscope, how they look like those chromosomes. Did you see it? This is so pretty. They are laid down in what we call in the middle of the cell. We call that the metaphase plate, the middle of the cell. Let me choose a different color. In the middle of the cell, this is what we call metaphase plates. They are laid down, the chromosome. Look at them. And each centromere for each chromatid sister face the pole, face the opposite. This here and this here. Very beautiful. It's wonderful. Science is something very pretty. The chromatid sisters are aligned in the middle of the cell. The middle of the cell, this is what we call metaphase plate. You are not going to miss this phase. And during this phase, this is how the chromosome look like. Remember this? If I give you this chromosome as a picture, immediately you are going to tell me that this is metaphase chromosome. This is the only phase you are going to see the chromosome as pretty as like that. Look at this centromere. This is one chromatid sister, and this is another chromatid sister, and this is the centromere, which is the neuro. This is the centromere. This is the neuro that will separate the chromosome on small segment and a long segment all the time. Small, long, and this is in the middle. That's your neuro, which is like a west, you know. This is your centromere. Separate the chromosome on two bras. We call that on two arms. Short arm, a long arm. Short arm, long arm. And this here is the centromere. Each chromatid sister has her own centromere. And this is the metaphase. They are very pretty. You are not going to miss how the chromosome look like during the metaphase. Okay, so let's keep moving. Did you see that? Our two chromatid sisters, look at them. They're laying down in the metaphase plate, right? And look at these spindles. We have, you got it. We have the kinetochore microtubule spindle, and this is non-kinetochore mitotic spindle. I'm going to tell you the difference between non-kinetochore microtubule spindle, the function, and kinetochore microtubule spindle. All right. Just critical thinking. Let us think what this microtubule kinetochore spindle are going to do over here. What they are going to do? They are attached to each chromatid sister. So they are going to pull them to the opposite pole. This is what they are doing. They are going to try to separate those chromatid sisters. And this is exactly what they are doing, guys, over here. This is exactly what they are doing over here. I don't know why I keep having. This is exactly what they are doing over here. They are pulling them on opposite pole. And those non-kinetochore microtubules, what you have to follow, you have to make, it's like an elastic and you are pulling. They are making the cytoplasm elongate. Those are going to separate the chromatid sister and those are going to elongate the cytoplasm. Of course, there's going to be two cells, two daughter cells. So they have to have a space, all right? So this happened during the anaphase phase. The anaphase phase, this is the separation of the chromatid sister. And then each chromosome now, we are not going to call it chromatid, they are separated. Each chromosome daughter move to the opposite pole. And in the same time, the cytoplasm is elongated by this non-kinetochore microtubules. So the kinetochore microtubules separate the chromatid sister, and then these chromosomes daughters are moved to the opposite pole. In the same time, the non-kinetochore microtubules are elongating the cytoplasm. What happened during the telophase? Look at that. Telophase. This mitotic spindle disappear. They are no longer there. and the daughter nuclei start to be formed. And look at that nuclear envelope. Look at those fragments. The one that we saw before. This is like the opposite of this telophase, just opposite. Just another, it's just like the opposite, it's going reverse, right? It coming over here to here. It's like going to form another, and nucleolus appear, is forming. And most of it also, the cytoplasm, you remember that it got elongated by the non-kinetochore microtubules? The cytoplasm is going to be separated by what we call cytokinesis. And this is by cleavage furrow for animal cells. For the plant cells, the cytoplasm also gets separated. We have almost the same things that happen, except one thing, very important for the plant cells. We do not have centriole. We don't have centriole for the plant cells. For the animals, without centriole, the cell cannot divide, cannot. We try it. I try it myself, as I told you. I spent 6 months with Philip Pahin learning about cell cycle, animal counts, and all this nice stuff. We knocked it down, the centriole, and the cell cannot divide. But with the plant cells, we don't have centriole, but they divide because they have other structures. To be honest, we don't know until now which structure, because the plant have a centrosome, but their centrosome does not have a centriole, maybe something else. And centrosome also is made of microtubules. So in the plant, so this is one thing, we don't have a centriole, but they divide the same way, and the mitotic spindles is a form from the centrosome, all right? And their cytokinesis, it's a form by a formation of the cell plate. Cell plate. They are vesicles forming from the Golgi, coming secreted in the middle, and the cell plate is a form, and therefore we have a new cell wall that separates two daughter cells. The animal cells is by cleavage furrow, which is a contractile ring of microfilament. All right, forming two daughter cells, and here is a cell plate forming from vesicle from the Golgi, making a new cell plate that will separate two daughter cells. So the division for the plant, it's almost the same things. As they said, we don't have, you see, we don't have a centriole. The prophase, the same thing. The prophase is condensing, nucleolus disappear, fragmentations of the nuclear envelope and the chromosomes are now visible, and they are two chromatid sisters. They're in the prometaphase. During the metaphase, we have the spindle is complete, and we have microtubules, kinetochore and non-kinetochore microtubules, the same thing. And then you will have the cell plate will form. Anaphase, the chromatid sisters are separated, right? And they are moving at the opposite pole. And then during the telophase, the daughter nuclei are forming. That's it. And cytokinesis began. the cell plate grow outward to the edge, of the cell. That's the cell plate. So now I want you to train yourself on identifying all the phases here. If you have any difficulty, email me and we can meet over here and they will show you. This, for sure, this is an anaphase. First of all, which type of cells is this? Is it animal or plant, and why? There are two things I can tell that this is a plant. First of all, the form is rectangular. Second is the formation of the cell wall, cell plate. Look at that. That's a cell plate. This is a telophase. This, you are not going to miss it. This is a metaphase. This is an anaphase, another anaphase over here. This, I will tell that this is a late prophase, but it's a pro-metaphase too. This is an telophase because I can see the cell plates appearing. This is a pro-metaphase. This is two daughter cells because the cell plate is newly formed. That's the cell wall. That's a prophase. It could be a late prophase. Late prophase. How the prokaryotic cells reproduce? They reproduce also by divisions, all right? But they reproduce by binary fission. The chromosome will replicate. This is the origin. It will replicate at the origin of replication. They have like 1 or 2 or 3 prokaryotic cells. Why of origin of replication? Why eukaryotic cells have thousands of it? And when we studied the gene replication, I will show you. I told you science is so pretty. So it starts at the origin of replication. He copy into copy, and he starts moving. And once he is at the opposite pole, that's it. He divide by binary fission. And the two daughter cells move apart. Of course, we have everything have to be under control, right? Everything should be under control in our body. Anything. Our sugar should be under control. Our metabolism should be under control. Our cardiac rhythm should be under control. Everything should be under control. You know? Cell cycle should be under control. What if the cell is not controlled? It starts dividing, going through like a machine. It's tumoral cells. Escape the control. We have a checkpoint. We have a checkpoint in the cell cycle. The frequency of the division, you know, vary with the type of cells. Not all the cells have the same duration of the cell cycle. Some of them divide quicker, some of them very slow. Most of it, they have what we call cell cycle control system. They have some specific chemical signal that they are present in the cytoplasm of the cells, or they are coming from outside to regulate both the internal and the external control. So they have like a checkpoint. So we have one at the G1. This is a checkpoint that will decide is the cells going to go through division or not. We have one that is from the G2 going to end phase. It's the G2, does it, okay. Is the cell normal to go through the division? Is everything being replicated correctly? And we have also an M point. if the cells, is it going to go outside or continue circulating? So when during the G1 checkpoint receive the go-ahead signal, which is the green light, okay? So the cell will go through the S phase, then the G2 phase, then M phase, and the cell divide. If it does not receive the go-ahead signal, what's going to happen to this cell? The cell exits the cell cycle and enter what we call G0 phase, which is like a resting phase. It's not dividing. You go to G0 and eventually it will die. We have also other that's sorry, that's the importance of checkpoint. We have also some regulatory factors or proteins that are involved in the cycle control. We call those cycling. We have what we call cyclin CDKs, cyclin dependent kinase. And we have also another type of factors. We call that maturations promoting factors, which is the CDKs complex that trigger the cell passage past the G2 phase. Show you in this. So we have the cyclin dependent CDKs, and we have the maturation promoting factors. The CDKs play a role. I mean, the activity of cyclin and CDK fluctuate during the cell cycle. During the cell cycle, fluctuate during the cell cycle. Look at that, during the M phase, more of it during the cyclin, M phase, the M phase. The MPF will play a role after the cell passes the G2 phase. Most of it promotes the mitosis peak during the metaphase of the mitosis. This is when it plays a role. It's the cyclin, the cyclin dependent kinase. It's like they are sending a signal for the MPF to be arise during the metaphase. I said earlier that the cell cycle is under internal and external signal from the cytoplasm. One of the internal signal, you know what is it? It's that the kinetochore, that they are not attached to the spindle microtubules, will send a signal to delay the anaphase. They have kinetochore that should be attached to the microtubules that should be attached to the, not all of them, but some of them. Supposedly, all the microtubules are not attached to the kinetochore. The cells, I mean, the chromosome cannot be separated, right? The chromatid sister will still attach. Well, the kinetochore are going to send a signal saying, hey, I need some microtubules attached to me so I can be able to separate my chromatid sister. Not all of them, because the non-kinetochore microtubules will play a role on elongating the cytoplasm. Supposedly, all of them are not attached to the kinetochore. Or supposedly, all of them are attached to the kinetochore. In any way, there are some internal signal coming, hey, something is wrong here. Come repair it. Some external signal is growth factors, and we realized when I studied in 1991, I think, or 92, I can't remember. I have a paper that discovered that one of the growth factors came from the milk of the mom. It was the top. I done an in vivo study about it. I found it like it's in the milk. It's the epidermal growth factor. At this time, when I studied the epidermal growth factor, they are like 7 members. Now they are like 22 members. And one of them is the trigger, the breast cancer. When I studied that, it was the top. And I published a paper about that. So some of the growth factors are released by certain cells that stimulate other cells to divide. Like, I mean, the milk from the mom, they bring growth factors to the baby so they can grow. For example, I give you an example over here. It's about the PDGF. PDGF, which is a plate-derived growth factors, stimulate the division of human fibroblasts. Here is, I'm taking human tissue, connective tissue, cut it in small pieces, making my cells, okay, homogenate, making it. It got attached to, I put it in a filter vessels that have fibronectins on it. It got attached. If I didn't put the media, in the media, some growth factor, this PDGF, he will grow very slow. Look at that. Barley 4 cells are growing very slow. But when I add in my media that I nourish the nutriment I am giving my cells in vitro to grow, I put on it this external growth factor. This is a skin, by the way, connective tissue. They start growing very nicely. Now, this is a normal cell. So what if I have tumor cells? Tumor cells, you see, I told you here about this human normal connective tissue. I should put them in a fibronectin vessels. I mean, these vessels are coated with fibronectin, so they can grow. They need a matrix. They need to attach to something. Tumor cells, they don't need a matrix. They don't develop any anchorage dependence. They don't care. You can grow them in glass. You can grow them. You take a piece of stone and you disinfect it and you making sure that it's clean and you put your tumor cells on it. They can grow, no problem. You put them in metal, sterilized, they can grow. You don't need a support matrix. This is why we call it does not exhibit anchorage dependence. Does not exhibit anchorage dependence. They should not be attached to substratum in order to divide. All right. Another example. Another example is clear. I will call you back. Another example is that I will tell you this. It's happened to me, I swear, at the VA hospital. I have my lab there, and I am growing cells that they said that they are normal. I grow them, and they work with them as normal cells. That means they are not tumors. We immortalize them. And one day, I didn't understand. I saw that they are growing in the top of each other. When they reach confluence, they grow in the top of each other. That's not characteristic of normal cells. It's like you are going in classroom, and the classroom ability is 30 students. That means you have 30 chairs, and each chair has one student. If I reach this capacity, I'm no longer going to accept a new student. The administration will stop. Even if the student asks, okay, I want to go with Dr. Carver. Oh, she don't have. She reached the capacity. It's the same thing. We reach the capacity. Where the tumor cells, they don't know that what's near reaching the capacity. They can grow until they reach completely the surface, and then they start growing in the top of each other. So they don't exhibit density dependent inhibition. They don't. Normal cells, they do. Once they reach the density dependent inhibition of the administration, what she do? She stopped adding students in my classroom. But the normal cells, when they get crowded, they stop dividing. They reach the confluence. And once they reach the confluence, I have to subculture them. I have to go and put them, like I divide them in small petri dishes. While the tumor cells, well, they grow in the top of each other, exhibit neither density-dependent inhibition or anchorage dependence. Look at this example. This is anchorage dependence. That's normal cells. They grow, right? And we receive the dependent inhibitions, all right? Density-dependent inhibition. They don't grow in the top of each other. Look at the tumor cells. They don't have that. Look at them. They grow on the top of each other. That's so funny. So the cancer cells do not respond normally to the body control mechanism. The cancer cells may not need growth factors to grow because they can synthesize their own. I mean, breast cancer cells, they overexpress growth factors. And they also have abnormal cell cycle control. They don't have those signal green, go, stop. They lost it. And tumor cells invade surrounding tissue by a process we call metastasis. That means exporting cancer cells to other parts of the body where they may form additional tumors. I mean, you can see intestinal-like cells in the brain, for example. The breast cancer cells are, since it's the month of October, and this is the month of cancer cell awareness. I mean, the tumor grow from a single cancer cells, one cells, escape, become abnormal, he can develop tumor, and if we catch it on time, we can stop the invasion. But if he goes stage 3 and 4, the cancer cells spread through the lymph and the blood vessel to other parts of the body. And the cancer cells can survive and establish new tumor in another part of the body. And by understanding, I mean, the cell cycle and how the cell cycle work and the cell cycle signaling, it's lead to an advanced cancer treatment. So now let's have a summary. So we have the cell cycle is 90% is interphase. Interphase, they have a gap 1 phase, G1, have a phase S, and have a gap 2, G2 phase. There in all these three phases, the cell continue to grow, but each phase is different function. During the G1, any structures inside of the cells is going to be replicated, except the DNA. The DNA is going to be replicated during the S phase, while the cells continue growing, the growth. The G2 phase is like a phase of controlling that everything is looking double, seeing double. And also the centriole that has been replicated during the G1 phase is going to start separating for animal cells, because centriole exists only in the centrosome of animal cells. And then the cell enters the M phase, which is the mitotic phase. They represent 10% of the cell cycle. The mitotic phase have a prophase, prometaphase, metaphase, anaphase, and the last one is telophase and cytokinesis. We describe each of those stages of those phases, and I really, really want you to be able, this is a plant cells, I really want you to be able to identify all those stages here, looking at this virtual slide from microscopy. And then I want you to get very, very familiar how metaphase chromosomes look like. This is two chromatid sisters attached by centromere, by their reciprocal centromere. This is one chromatid sister with the centromere. This is another chromatid sister by the centromere. They are attached by their centromere. Okay? This is very nice. When you look at this is chromosome from a metaphase. And the centromere separate the chromosome from the short arm and long arm. We arrive at the chapter. As I said, the number of chapters may differ from each book, which book you are using, but don't rely, but rely more on the title of this chapter. So this is chapter 11. It could be chapter 8 for some. It could be chapter 7 for some. But most of it, we are going to talk about mitosis and sexual reproduction. If you look at this girl, this is an actress. I forget the name. She looked like her mom, and this one looked like her dad. So before we start any early, before we go any further, we still have like 15 minutes. What is the genes? The genes are made up of segments of DNA, right? And they are the unit of heredity. Genes are passed often to the next generation via cells that we call gametes, sperm for man and eggs for female, right? Each gene has a specific location, and those locations we call it locus. Location, locus on a certain chromosome. Most of the DNA is a packed into a chromosome. We know that, and when we add to these chromosomes proteins, we call it chromatin. In a sexual reproduction, a single individual passes genes to its offspring without fission of the gamete. This is asexual. And this generation, the offspring generation, they look like completely from the parent. This is a clone, identical clone. They are genetically identical individual from the same parent. The sexual reproductions, however, the two parents give rise to offspring that have a unique combination of gene inherited from the both parents. So you have to know that the human somatic cells, any other cells than the gamete cells, than an egg, have a 23 pair of chromosomes, which mean 46 chromosome, all right? And the 2 chromosomes in each pair are called homologous chromosome or homologs. The homologous chromosomes is very funny. They have the same length and the same shape and carry the gene at the same locus. If this locus, this is a gene for the hair, in parallel, the same locus is for the hair. It doesn't matter if the hair color is brown or the other one is red, but this is the locus gene for hair. Same, it's parallel. So they carry the same length, the same shape, and carry the gene controlling this inherited character. The 6 chromosomes are the one that will determine the sex of the individual, are called X and Y, those chromosomes. And the females have a homologous chromosome pair of X, XX, and the male have 1 X and 1 Y chromosome. The remaining 22 pair of chromosomes are called autosomes. Each pair of homologous chromosomes include 1 chromosome from each parent. The 46 chromosomes in human somatic cells are 2 sets of 23, 1 from the mom and 1 from the father. The diploid cell, which is so much information, 2n has 2 sets of chromosomes. For human, the diploid number is 46, therefore 2n equal 46, n equal 23. The sperm cells, the sex chromosome may be either X or Y, right? And the egg, it's not fertilized yet. The sex chromosome is X because, as I said, the human eggs are homologous pair of X chromosome, XX. So therefore, the unfertilized egg, the sex chromosome is only X. The male, the sperm, should be either X or Y, because the human male have X and Y chromosome. When fertilizations happen, when a sperm enters the egg, we call that a zygote or fertilized egg. Zygote. And has one set of chromosomes from each parent. The zygote produces the somatic cells by mitosis and develop into an adult. Who produced those gametes? Those XX and XY. The ovaries produce the XX and the testicles produce the haploid gamete. Gametes are the only type of human cells that's produced by meiosis. So anything inside of your body is by mitosis except your sexual cells. Meiosis results in 1 chromosome set in each gamete. Fertilization and meiosis alternate in sexual life. So this is exactly mitosis and meiosis alternate. The alternations of meiosis and fertilization is common to all organisms that reproduce sexually. ovaries test, right? You go through meiosis, the testicle produce sperm, haploid, and egg haploid. This is all N. Once they fertilize, they merge, they form double N plus N, double N. They form a zygote. Zygote will develop and divided by mitosis to produce organism, which is the organism is double N, diploid. Gametes are the only haploid cells in animals. This is the only haploid cells that we have in our body also. They are produced by meiosis and there go no further divisions before fertilizations. When they get fertilized, they form a zygote that divide by mitosis to develop into a multicellular organism. So by definition, meiosis is a type of cell division which reduces the chromosomes number. It's type of cell division. It's sexual cell of division, right? It's only happen to the organism that sexually reproduce and reduce the number of chromosome of the parent cells by half. Look at that. This is double N become N. Double N become N. So that's true. And involve two successive divisions during the meiosis. We have meiosis 1 and meiosis 2. So we are therefore going to study the stage of meiosis now. We are going to study the stage that will give me those cells. This meiosis, we have two types, meiosis 1 and meiosis 2 over here. Remember that I told you that the chromosomes are by chromosomes homologs, right? During the meiosis 1, homologous pair up and separate. So the objective of meiosis 1 is to separate the chromosome homolog that has been replicated to chromatid sister, all right? So the chromosome homolog that line up are going to be separated, and each chromosome homolog has already chromatid sister on it. In the meiosis 2, the objective is to separate those chromatid sister. The result will have 4 haploid cells that they are not identical to each other, not identical to the parent. Not identical to the parent for sure, because they are haploid, they are not diploid. The parent is diploid. For sure, they are not identical to the parent. But to not identical to each other, this is the key. This is what we are going to study right now. So let's look at the meiosis. Before the meiosis 1, I have to go through the interphase, because I told you any cells before to get replicated have to go through interphase. And during this interphase, I have G1 initiated cells. They have structures on it. Right? It's an ovary or testicle cells. You have structures on it. You have also Golgi, everything. So everything gets been replicated except the chromosomes. G2, S phase, chromosomes are going to be replicated. So imagine that this is my cells and have a 2 chromosome, pair of homologous chromosome. There 2. Same shape, the same length, and have the same genes. Focus. All right. S phase, they get replicated. All right, they get replicated. So now I have chromatid sister of this blue chromosome, chromatid sister of this homologous chromosome. All right. So the meiosis 1 will separate those homologous chromosomes. It will separate them. Now I have 2 haploid cells with duplicated that separate. Each chromosome homolog go to one cell. But they still chromatid sister. Each chromosome they are haploid, but they have chromatid sister. Now I need to separate those chromatid sister. This is going to happen during the meiosis II. I've done it very bravely, but it's more complicated than that. Remember one thing, that meiosis I is always preceded by an interphase. If you realize it, meiosis II take over as soon as the telophase I finish happen. I don't have interphase before the meiosis II. I don't. Immediately I have the meiosis II happening. So let's look at now very closely. So during the meiosis I, imagine that interphase already happened. I have prophase I, prophase for meiosis I, metaphase I for meiosis I, anaphase I for meiosis I, and telophase and cytokinesis for meiosis I. Okay, so let's look at very briefly what's happened. Stay with me. Okay. I start with 6 chromosomes. My mother cells. The mother cells have 6 chromosomes. First of all, I remember in the G2 phase, what's going to happen? Chromosome homologue are going to align with each other. For some reason, it's so funny. They are going to go, okay. You see how the chromosome homologue are already aligned here. They're in the prophase one. They are already there. This happened actually during the G2. They go short, the middle length will go with the middle life. The small one will go this small one. The long one will go this one. They align with each other. And each chromosome homologue is already being duplicated during the S phase of the interphase. Correct? And they enter the prophase one. What's going to happen in the prophase one? Fragmentation of the nucleus, the envelope nuclear, envelope fragmentations. The nucleolus, you cannot see it, is gone, right? And you have those centrosome where a pair of centriole that has been separated during the G2. The same thing that's what happened during the mitosis, right? This starts moving to the opposite pole, forming this spindle, mitotic spindle. Same thing, they're moving, right? One thing I want you to notice very, very well. It's so extraordinary, wonderful. And that might explain why you don't look like your sister or your brother, and you don't look like your dad or your mom, but a mix of it. During this, when there are this alignment of chromosome homologue with each other, they are so close that they are going to exchange materials. They are going to exchange materials. They form what we call chiasmata. Very pretty. They have some synapse happening here. Oh my God. Did you see that? Isn't that beautiful? In the synapse, homologue chromosomes are loosely paired up with each other, aligned gene by gene, and they exchange material. Hey, you blue, give me some blue, I give you some red. They done it. During this chiasmata, this synapsis, chiasmata. Of course, chromosomes began to condense. Right? During the metaphase one, the mitotic spindle already well beyond, very wonderfully formed. Centriole in each pole, beautiful. Again, we have this non-kinetochore microtubules, kinetochore microtubules, kinetochore microtubules, non-kinetochore microtubule. Remember the difference, right? If you don't, go back to the mitosis. I want you to go and look at it, please. The difference between kinetochore microtubules, non-kinetochore microtubules. One thing is very beautiful. You are going to see my chromosome homologs, that they are paired with each other, right? And each chromosome is a pair of chromatid sisters that are going to lay down in the middle of the cell, in the metaphase plate. It's not that pretty. In the anaphase, I'm going fast. Let's go back over here to the prophase. During this chiasmata, they are lining down. I just forget this keyword. It's cross, and you know when they exchange this material, hey, red, give me some blue, and the blue, give me some red. Exchange materials. It's happened between non-chromatid sisters, none, but between chromosome homolog, all right? This is what we call crossing over, exchanging of DNA fragment during the prophase 1. And they form a tetrade. Look at that. 1, 2, 3, 4. 1, 2, 3. It's a tetrade. A group of 4 chromatid. And this is a crossing over because we have an X-shaped regions where the cross happen. This is an X. We form an X. Where the cross happen, we have like an X. See? We don't have it here. It doesn't happen over here. As we happen here, we have an X crossing over. We have an X shape. I just forget that, sorry. So let's move on. So we got the metaphase, tetrad line up at the metaphase plate, right, with one chromosome facing each pole, and each chromosome homolog have chromatid sisters still attached to each other. Microtubules from one pole are attached to the kinetochore. Microtubule from the other pole are attached to the other kinetochore for the other chromosome. All right, and then during the anaphase, you got it. They are going to separate the chromosome homolog. One chromosome homolog move toward one pole, and the other chromosome homolog move toward the opposite pole. Sister chromatids still attached. Look at that. Still attached to each other. But how are those chromatid sisters the same than those chromatid sisters I start with? No, because they exchange. They have some genes them that don't belong to them. It's not that pretty. And chromatid sisters still attached to their centromere, right? And the telophase and cytokinesis. In the beginning telophase, each half of the cells has an haploid set of cells. Look at that. 1, 2, 3. 1, 2, 3. We start with 6. We end up having 3 on each cell. 3. And each chromosome consists of chromatid sisters. And of course, cytokinesis happens simultaneously, forming 2 haploid cell daughter cells. Why they are haploid? Because they are half of the number of chromosomes. 1, 2, 3, 1, 2, 3. So this is an N, because I start with double N equals 6. 1, 2, 3, 4, 5, 6. And then I have 3 red and I have 3 blue. So I have 6. Double N equals 6. Here I don't have double N. I have only N. I have 3 because N equals 3 over here. In animal cells, the cleavage ferro forms. This is animal by the cytokinesis by cleavage ferro, remember? The plate forms for plant cells because we do have some plant that also divide by sexual reproductions. since the chromosome already been replicated into chromatid system, we don't need an interphase. We don't need an interphase to replicate again whatever that had already been replicated prior to meiosis 1. So therefore, we enter meiosis 2 immediately. Meiosis 2, we have prophase 2, metaphase 2, anaphase 2, and metaphase 2 and cytokinesis. And meiosis 2 is very similar to mitosis for the simple reason that the objective is to separate the chromatid sister. So remember, I end up having two cells. Each cells have a chromosome 3 chromosome homologue. And each chromosome homologue is already replicated on chromatid sister. They are not identical to each other, those two cells. Look at that. So I have the prophase 2. Remember the nucleus disappear, fragmentation. Centrioles are going to be moving toward the opposite pole, forming the mitotic spindle. Kinetochore, non-kinetochore. During the metaphase 2, the chromatid sisters are going to lie down in the metaphase plate, all right? And because of the crossing over that's happened during the meiosis 1, during the prophase 1, the two sister chromatids of each chromosome are no longer genetically identical. They're not. The chromatid sister are not. Look at this chromatid sister, doesn't have any blue on it, and this one have a little blue on it. This one have a little blue, this one no, this one have a little red. So it's completely genetically different from each other. The kinetochore of sister chromatid attached to the microtubule extending from the opposite pole. And then anaphase 2 happened. The sister chromatid has been separated, all right? And the sister chromatid of each chromosome now move as two newly individual chromosomes toward the opposite pole. And the telophase 2, the chromosome arrive at the opposite pole, the nuclei start, and the chromosome began the countdown scene. And cytokinesis, this is an animal cell, so it happened by cleavage Pharaoh. And each identical cells, its daughter cells, is genetically different from each other and from the original parent, and have half of the cells. So we end up having 1, 2, 3, 4 haploid cells, not identical to each other, and very obviously not identical to the parent. So if I want to compare mitosis to meiosis, the first thing I can tell that the mitosis conserve, because he gives two daughter cells identical to each other, identical to the parent. If your skin is Renault dividing, you will have the same type of skin, right? Identical to the origin. So mitosis conserve the number of chromosomes set first of all. If I come from double diploid, I will end up with diploid, producing cells that are genetically identical to the parent, identical to each other and to the parent. Meiosis, however, reduce the number, not only reduce the number of chromosomes set from 2 diploid to haploid, producing cells that they are differ genetically to each other and differ to the parent. And over here, I just put a table that I want you to go look at it by yourself. Differentiation between mitosis and meiosis. And that's end our lecture for today. Thank you so much, and I will see you next time.